Hi, this is Fredrik Wermling again. Thanks for watching this video. Today, in this video, we're going to talk about analyzing CRISPR screens using cBioPortal. Once again, a super impressive database with so much data in there. And as you know, this is part of a list of videos where I describe different tools that could be used to analyze CRISPR screen data. And cBioPortal contains a lot of clinical studies of different tumors. You see how many samples there are, what type of cancer they are related to. You can home in on a particular cancer type, or you can go a little bit broader, sort of pan-cancer studies. You see up here that there are 497 studies available, 329,000 samples. As alluded to earlier, some of these databases or tools get overwhelmed when you're trying to do too complicated analysis. I think there are sort of ways to use this type of data as an API or so, accessing the data in different ways. And with that, I'm just suggesting that don't try to analyze 329,000 samples because it's gonna crash probably, or most probably. But if you're interested in sort of a bit broader analysis, I would suggest to use this TCGA Pan-Cancer Atlas that has 32 studies. You can see here they all will be called Pan-Cancer Atlas, and here's 92 samples, and then it will sort of include some representative samples from most cancer types. So there's also a curated set of non-redundant studies, but then this is very big, so it's going to take a long time. Oftentimes I start with this Pan-Cancer Atlas, and in this case you can explore these studies. So clicking this one I encourage you to do. Then you can go into the data and sort of look at the whole study, not just focusing on certain genes. But in this case, since we are analyzing CRISPR screen data, I'm going to choose this setting, and I'm going to take the A549 genes, possibly enriched genes, this shorter list. Again, I'm doing that intentional. I don't want to have too many genes here because it's going to take longer time. But of course, if you have a longer list of genes, you just have to wait. But I think for the purpose of this video, it doesn't make any difference. So I paste them here. I can select some sort of data that I want to be included here, just so you're aware of it. I'm not going to make any changes to it this time, and then I press run here then. And then you'll see that it's sort of it's creating 16 genes in its 10,967 samples. One of the main data sets that you're getting out from here, which I think could be perhaps one of the more relevant outputs at this stage in the CRISPR screen, is this oncoprints. You have several tracks here. You can modify which ones you want to have here. You can download them in different ways, both as numbers and images, etc. It takes a little bit of a time to sort of understand these ones. This is study of origin with different colors. Down here you can see the different colors and what they are representing. So they're essentially representing these different cancer studies, right? So a different color means a different cancer subtype. In this case, there's 32 of them, so the colors are difficult to follow. But I'll give you an example later on how you can dig into this a little bit more. And then if I go like this, you see each line here will be, in this case, representing 6 patients. You can zoom in more, so you get to a level where sort of you see individual patients. By scrolling here, you can move around in the data set. So now it becomes a little bit more, higher resolution essentially for what you're looking at. But then what an oncoprint is doing, and I think it's very useful type of data. I think when you're not used to it, it's however, it could be a little bit difficult. But essentially here, now you know that when I hover over like this, you see a line, and in this case, each line will then represent 7 patients. And then you can look for each of these genes, if they have a mutation, and which type of them. And then they're typically sorted like this. So you'll see here, TP53, 36% of these cancers have mutations in them. You'll see that they have different types of colors here. And again, colors in this case, you have to look at here, gene alteration. So there could be missense mutations, putative driver. A common one is a green one. Black one, I think, is truncation mutation. A putative driver, you have a deep deletion, amplification is red, etc. And what you can see then is in this data set, you can see how many of these genes are mutated. So you can look at your CRISPR data set. Are there any of these genes that commonly are mutated, and think about, does that mean anything? I think it could have a diagnostic relevance, right? If your CRISPR screens show that you see an enrichment, for example, cells with this mutation, and you see, for example, that a lot of cancers are mutated in this, that could be relevant. Although, of course, even if it's very few, that could still be relevant, right? But this is the type of understanding you can get out from this. You can see what type of genetic change there typically are. So here for PDCD10, for example, 5% of the cancer have mutations, and most of them are actually this red one, so they're amplified, while P10 most of the time have these deep deletions of the mutations. You can also identify samples then that have combinations of different mutations or that are single mutated. So here are a set of patients that only have p53 mutations. Here are ones that have both p53 and P10, for example. So I think this could be a very useful thing to get an idea about your particular gene set, and if they are they mutated or not in cancer? Are they relevant cancer genes, for example? And then there's so much you can do here, and I'm just going to touch upon a little bit of this. So we have the cancer type summary here, which I think could be very useful. And then you can see the alteration frequency, and now I highlighted all these genes. So uterine carcinosarcoma, have almost 100% of them have mutations or amplification or multiple alterations, as shown here for all of these. And then you can go down and see some of these have much less of it. Maybe this is not super relevant information, but it could be very interesting. If it turns out that you're interested in one of these genes, you can start understanding, okay, if I'm interested in this gene, which of the cancers are more relevant to look into? And for example, lung squamous cell carcinoma, you have more than 30% of them showing amplifications of these. So if you're interested in this, maybe this is the cancer type you should look into. While, for example, kidney, renal papillary cell carcinoma has very little alterations, so this might not be as relevant, right? So this becomes the way of sort of screening into this if you want to go deeper. A very cool but also very dangerous way of analyzing here is is this comparison type of data. You can look at and make really nice looking survival curves based on mutations in your genes. So you can press survival here and see if you have mutations in any of these, or if you don't have any changes, do we see any differences in survival of these different groups? And for example, here you see really strong p-values. So having these mutations, looking at it just like this, it looks like having mutations in these genes is bad prognosis, right, compared to unaltered. However, just as they say here, interpret these results with very much caution. Because and this is especially true when you start including different cancer types, as we do here. We have 10 or 32 different studies. There is a lot of different cancers. So what we probably are doing here by selecting for patients that have mutations is not necessarily that we're looking at, or is this mutation contributing to the survival? We are probably more likely, what we're looking at here at least, looking at that we're selecting for certain cancers that have these mutations, but it doesn't mean that it's a causative link, right? And I think you can look at, if you look at clinical data here, for example, you'll see that this type of analysis, you're biasing the data into different cancers. So here in the altered group, you'll see that there's a strong difference in just which cancer. So you essentially don't do this type of analysis in this way. I think if you want to do the survival curves and do it based on genes like this, I think you should go for one defined diagnosis or one clinical cohort, or at least just focusing on one type of cancer. So you're looking at a particular type of lung cancer with or without the mutation. But again, it looks pretty nice, and it's very easy to click around and sort of generate this data set, but be careful about them. Yes, this is cBioPortal. Again, I think the OncoPrint could be a very good starting point for you to sort of look into a wide set of cancer types and seeing, is there any of these genes that are relevant for these cancers, or at least they're mutated? How are they mutated? What type of mutations are? And that makes it possible for you to sort of go into them in more detail later on and sort of focus on different type of cancers if you're agnostic to that at the starting point. So with that, I thank you for watching this video. I hope you learned something, and as you know, there are more videos in this series and that you can look at, so please do that if you're interested in, and have a great day.